Welcome back, everyone. Today we're taking a deep dive into extremophiles and alternative biochemistry. We've got some fascinating research articles to unpack here. Our goal today is to really push the boundaries of what we think we know about life's limits.
This collection of research really makes you reconsider some of those basic assumptions we have about where life could begin and what shapes it might take. We're really going beyond Earth-based biology here.
Absolutely. And one of the things that really jumped out at me was this idea of the extremophile paradox. Ian von Hegner lays this out in his paper. It seems strange that organisms thriving in such brutal conditions, you wouldn't think that they could have evolved in those very same places.
Yeah, it's a really easy point to miss. The paradox kind of highlights the crucial importance of environmental history. Extremophiles, as hardy as they are, they actually come from less specialized ancestors. We call these mesophiles, and these guys lived in much milder environments. Over a really long period, these mesophiles gradually adapted to these harsher niches, and that led to the extremophiles we see today.
So even the toughest extremophile had a more normal ancestor. But how does this paradox actually change how we look for life beyond Earth?
Well, it changes the way we think about where to search. Imagine a planet that's always been uniformly extreme. So even if we found organisms that could live there now, the paradox suggests that life couldn't have started there, it couldn't have originated. But on the other hand, if you have a planet with a variety of environments, some mild and some extreme, that offers a more plausible way for life to evolve and branch out.
So a planet with a history of different environments might be a better place to look, even if it has some pretty crazy conditions right now. It's not just about habitability now, but how the environment has changed over time.
Exactly. And this kind of brings us to another cool concept that von Hegner mentions, the hopeful monster. This is like a hypothetical organism that makes a sudden evolutionary leap. It jumps right into an extreme environment without going through that gradual adaptation process we usually expect.
It's a real challenge to the idea that evolution always happens slowly, step by step. Is this hopeful monster idea even realistic, or is it just a fun thought experiment?
It is improbable, but it's not impossible. Such a scenario would depend on a very specific and unlikely combination of genetic mutations. But the fact that we can't rule it out completely raises some interesting questions. What if a hopeful monster could just skip the need for slow and steady adaptation? That might mean that life could arise in places we thought were just too harsh.
Almost like winning the cosmic lottery of evolution. But instead of money, you win the ability to live in, like boiling hot springs or an acidic lake.
Right, and that brings us to the bigger picture for exobiology. If we consider the extremophile paradox and the potential, even if it's slim, for these hopeful monsters, it really widens our search for life beyond Earth. We can't just focus on planets that look like Earth today. We got to think about their environmental history and these less conventional ways that life could have evolved.
Okay, so we're seeing how the origin and evolution of extremophiles gives us clues about where we might find life elsewhere. But let's shift gears for a minute and talk about alternative biochemistry. This paper, Solvent Compatibility with Biopolymer Folding and Evolution, Assessment for Alternative Biochemistries, really gets into this.
Yeah, this one tackles a fundamental question. Could life exist using solvents other than water? It's so easy for us to think of water as essential for life, but that's because our whole understanding is Earth-based.
Right. Basic biology teaches us that water is the universal solvent. It's crucial for all of those important biochemical reactions. But is it really universal? Or is it just universal to us?
That's the core of this research. The authors look at a whole bunch of solvents and evaluate how good they are for biopolymer folding and evolution. They've even developed a way to measure this, the biopolymer solvent score, or BS score. This helps determine how favorable a solvent is for life.
So it's like a rating system for how life-friendly a solvent is. Does a higher B score mean a solvent is better than water?
It's a bit more complex than that. The BS score lets us compare different solvents based on their physicochemical properties and how those properties affect the formation and function of complex biomolecules. Water gets a very high score, but other solvents, like formamide, for example, show some surprising potential.
So we haven't actually found life that uses anything other than water, but this research shows that it's at least theoretically possible.
Exactly. It really expands our thinking about what life could be and where it might exist. Imagine a world where life is based on formamide or methane instead of water. That has some major implications for exobiology.
It opens up a whole new world of possibilities. And speaking of expanding our horizons, let's talk about the concept of expanded genetic alphabets. Now, we're not just talking about different solvents, we're talking about different building blocks for life itself.
This is where things get really interesting. Xenonucleic acids, or XNAs, are essentially genetic systems that use building blocks other than the familiar A, T, G, and C nucleotides that we see in DNA. They use non-standard nucleotides, so they expand the genetic alphabet, and they might even boost the information carrying capacity of the genetic code.
So it's like adding a bunch of new letters to the DNA code. But does that even work? Can life actually function with an expanded genetic alphabet?
Research suggests it can. Systems like AGIS, which stands for Artificially Expanded Genetic Information Systems, have been developed to incorporate these non-standard nucleotides, and the results have been promising. These expanded alphabets can lead to greater information density and even brand new functionalities that we wouldn't see with a standard DNA code.
That's incredible. It means that there could be organisms out there with genetic codes far more complex than anything we've ever seen.
Yeah.
But how do we even begin to study these systems? It's not like we can just sequence XNA with our regular DNA sequencers.
You're right. Traditional sequencing methods can't handle these non-standard nucleotides. But that's where nanopore sequencing comes in. We've got a paper here that highlights just how capable this technology is.
Nanopore sequencing is already changing the field of genomics. It allows us to sequence DNA and RNA directly without having to amplify or label them. It's like we can read the genetic code in real time. But can it really handle XNA with those non-standard nucleotides?
That's the great thing about nanopore technology. Its direct reading approach and its single molecule resolution make it adaptable to a wide range of genetic material, including XNA. It can detect those tiny differences in molecular structure that make non-standard nucleotides unique, giving us a window into these previously inaccessible genetic systems.
Wow, it's like having a whole new way of seeing the hidden world of expanded genetic alphabets. But before we get too carried away with XNA, let's switch gears to a celestial body that's always captured the imagination of those looking for extraterrestrial life, Titan.
Yeah, Titan is a really intriguing mix of the familiar and the alien. Its thick atmosphere, rich in organic molecules, reminds us of the early Earth. But its surface is freezing, with lakes of liquid methane and ethane, a stark contrast to our planet.
It's a place that seems both alien and strangely inviting. And it raises this big question: could life, maybe very different from our own, exist on this mysterious moon?
We have this research paper, Towards Prebiotic Chemistry on Titan, Impact Experiments on Organic Haze Particles, and it offers some interesting clues. The study focuses on the impact of, impacts on Titan's organic haze. Turns out, while we usually think of impacts as destructive events, they could also be catalysts for creating the building blocks of life.
Wait, so you're saying asteroid impacts, which we usually associate with, catastrophic events, could help kickstart life?
That's exactly what this research suggests. The intense pressures and temperatures created during an impact can trigger chemical reactions. And these reactions can lead to the formation of complex biomolecules, things like amino acids and nucleobases. In a way, these impacts act like a cosmic crucible, forging the ingredients for life from the raw materials in Titan's atmosphere.
That's amazing. So even in this seemingly harsh environment, with its frigid temperatures and methane lakes, there could be processes at play that are setting the stage for life to emerge.
That's what makes Titan such a captivating target in our search for extraterrestrial life. It gives us multiple possibilities, and each one is more fascinating than the last. And it really emphasizes the need to expand our thinking when we're contemplating the potential for life.
It's like Titan is a cosmic mystery box, and we're just starting to peek inside. You know what? I think we need a moment to process all of this. We've covered a lot, and I think a little break is in order before we jump into the next phase of our exploration.
I agree. Let's give these ideas some time to settle before we move on. What's really interesting about the research on Titan's impacts is how it challenges the assumption that liquid water is absolutely essential for life.
It really is a paradigm shift, isn't it? We've been so focused on Earth that we assume what works here has to be the blueprint for life everywhere else.
And while water is definitely crucial for life as we know it, the possibility of life emerging and evolving in environments without water is a really exciting thought.
Titan, though, presents a pretty unique challenge with its super low temperatures. How can we even think about life in such an extreme environment?
Well, that's where our understanding of extremophiles comes in. Remember, we've talked about organisms that thrive in conditions that would be lethal to most life. There are extremophiles on Earth that can tolerate incredibly cold temperatures, like way below the freezing point of water.
So while Titan's surface might be frozen solid, there's still a chance that life could exist in those subsurface environments where the temperatures could be a bit warmer.
Exactly. And it's possible those subsurface environments could have liquid water kept warm by internal heat sources, things like tidal forces or geothermal activity.
So, we could have extremophiles swimming around in a hidden ocean beneath Titan's icy shell.
It's a compelling idea and one that scientists are actively investigating. But even if there isn't a hidden ocean, the research on impact-driven organic chemistry suggests the building blocks of life could be present on Titan's surface.
So, even without a subsurface ocean, there's still a chance that Titan could be harboring some form of life. Maybe one that uses those impact-generated biomolecules in ways we can't even imagine yet.
That's what makes Titan such an intriguing target in the search for extraterrestrial life. It presents us with multiple possibilities, each one more fascinating than the last. And it really highlights the need to broaden our perspectives when thinking about life's potential.
It's like Titan is this cosmic mystery box, and we're just starting to open it. Let's shift back to those expanded genetic alphabets and the potential of XNA. This idea feels both futuristic and like it's fundamentally important to how we understand the possibilities of life.
XNA really challenges us to rethink what biological information is. We've been so focused on DNA and RNA, assuming those are the only molecules that can carry genetic instructions.
But now we're seeing that there might be a whole universe of different genetic systems out there, maybe with much greater complexity and functionality than our own.
Imagine a genetic code that doesn't just have 4 letters, but 6, 8, or even more. The potential for encoding information and creating new biological functions would just explode.
It's mind-blowing. But wouldn't those complex genetic systems be super fragile? DNA is already vulnerable to damage from radiation and other environmental stuff. How could XNA-based life survive out there in the harshness of space?
That's a good question. The stability of XNA is a big area of research right now. It's possible that XNA-based life might exist in places that are protected from those harmful factors, maybe deep underground on a planet or a moon.
So those extreme environments that we thought were hostile to life might actually be the perfect places for these exotic genetic systems to thrive. It's really turning our usual assumptions upside down.
Exactly. And as we get better at detecting and analyzing XNA, we might start finding evidence of these alternative genetic systems in the most unexpected places.
This brings us back to the importance of nanopore sequencing and its ability to read those non-standard nucleotides that make up XNA. It's a tool that's opening up whole new frontiers in our understanding of life's diversity.
Nanopore technology is revolutionizing how we study genetics, both here on Earth and potentially beyond. Its ability to read long stretches of genetic material without the need for amplification or special markers is a game changer for exploring the full spectrum of genetic systems, from the familiar to the truly alien.
It feels like we're on the edge of a whole new era of discovery in biology and exobiology. The tools are getting more sophisticated, and our understanding of what's possible is constantly expanding.
It's a really exciting time to be working in these fields, and as we continue to explore the limits of life, I have no doubt that we'll uncover even more surprises that will challenge our current understanding and force us to rethink our place in the universe.
Before we get lost in the wonders of XNA and the search for life beyond Earth, it's important to remember what we know and what we don't about alternative biochemistries. We need to be careful not to get carried away with speculation.
You're right. Research on alternative solvents and biopolymer compatibility is still in its early stages. We have some theoretical models and promising results from the lab, but we haven't found definitive proof of life that uses anything other than water.
So, while the idea of silicon-based life, for example, is captivating, it's still purely hypothetical at this point.
Exactly, and that's an important distinction to make. While we should absolutely explore these possibilities and keep an open mind, we need to stay grounded in the evidence.
Which means we need more research, more experiments, more observations, more data. That's how we'll understand the true potential of alternative biochemistries.
That's the essence of science. Constantly questioning, testing, and refining our understanding based on what we observe. And even if we don't find silicon-based life or organisms with expanded genetic alphabets, just exploring those possibilities will deepen our understanding of life itself.
It reminds us that sometimes the most profound discoveries come from asking the most audacious questions. And when it comes to exobiology, there's no shortage of audacious questions.
Absolutely. And one of the biggest questions we can ask is, What are the true limits of life? What are the fundamental rules that govern how it arises, evolves, and diversifies?
That's a question that fuels our exploration of the universe. It makes us push the limits of our knowledge and imagination. And who knows, maybe the answer is waiting for us in some unexpected corner of the universe.
Speaking of unexpected corners of the universe, maybe this is a good time to revisit the possibility of silicon-based life. While it's still speculative, it's a concept that continues to fascinate scientists and sci-fi fans alike.
I'm all for revisiting it. I've always been intrigued by the idea of silicon-based life forms. They seem so alien, so otherworldly. But what are the scientific arguments for and against their viability?
Well, let's start with the potential. Silicon, just like carbon, can form 4 bonds, which is crucial for creating the complex molecular structures that are needed for life as we know it. And silicon is super abundant. It's the second most common element in Earth's crust, so it's probably plentiful on other planets too.
So, in terms of basic chemistry at least, silicon seems like it could be a contender for the foundation of life.
That's the optimistic take. But there are some big challenges to the silicon-based life idea. One major hurdle is that silicon loves to bond with oxygen. It forms these compounds like silica, which are really stable and don't react easily.
So even though silicon has the potential for complexity, it might not be as readily available or as versatile as carbon in most environments.
Exactly, and that's a key point. Carbon's ability to easily form and break bonds with other elements makes it such a flexible building block for life. Silicon, on the other hand, tends to get stuck in these stable, inert compounds.
But couldn't there be environments, maybe somewhere out there in the vastness of space, where silicon is more accessible and reactive? Environments where it could play the role that carbon does here on Earth?
It's possible, but it's probably a long shot, and that's what makes exobiology so captivating. It forces us to question our assumptions and consider possibilities that might seem outlandish at first.
So even though we don't have evidence for silicon-based life right now, we can't completely rule it out. It's still a fascinating possibility, a reminder that the universe is full of surprises and that what we know about life might be just a tiny fraction of what's really out there.
And even if we never find definitive proof of silicon-based life, the very act of exploring this idea will teach us more about the conditions needed for life to arise and evolve. It's a journey that's bound to lead to new discoveries and insights. But let's step back from the hypothetical for a moment and focus on something a bit more concrete. How can we use what we know about biochemistry to better understand the possibilities of alternative biochemistries?
I like that approach. Let's ground our speculation in what we already know.
Exactly. Let's take photosynthesis, for example, which is a fundamental process for life on Earth. Plants and other photosynthetic organisms use sunlight to convert carbon dioxide and water into energy-rich sugars, releasing oxygen as a byproduct.
It's the basis for most life on our planet. But how does this connect to alternative biochemistries?
Well, imagine an alien world where the main energy source isn't sunlight. It could be geothermal heat or chemical gradients.
So instead of photosynthesis, we might have organisms that use those other energy sources to power their metabolic processes.
Exactly. And those processes might use a completely different set of chemical reactions and biomolecules compared to what we see in photosynthesis here on Earth.
That's fascinating. By looking at familiar biochemical processes, we can start to imagine how life could function under vastly different conditions. But those analogies can only take us so far, can't they? We still have to consider the possibility of completely new biochemical pathways that we haven't even thought of yet.
You're absolutely right. And that's where the real excitement lies, in the unknown unknowns, the possibilities that we haven't even dreamed of. But even as we venture into those uncharted territories, what we know about biochemical processes will be a valuable tool. It'll guide our explorations and help us make sense of what we find.
It's like we're explorers sailing into uncharted waters. We have maps of familiar lands, but we know we're likely to find islands and continents that have never been mapped before.
A perfect analogy. And as we navigate those unknown waters, it's crucial to remember that the journey is just as important as the destination. Every new insight, every surprise discovery expands our understanding of life's possibilities. And it brings us closer to answering that fundamental question, are we alone in the universe?
On that note of cosmic exploration, I think we're ready for the final part of our deep dive. We've covered a lot of ground today, from the unexpected origins of extremophiles to the mind-blowing possibilities of alternative biochemistries. It's been a journey that's challenged our assumptions and sparked our imaginations.
It really has. And as we move towards the end of our discussion, I want to take a moment to summarize what all of this means for exobiology research going forward.
I'm ready. What are the key takeaways for researchers as they continue to explore the possibility of life beyond Earth?
Well, I think one of the biggest things to take away from all of this is that we need to broaden our definition of habitable. We've been stuck on finding planets that look just like Earth does now.
I get it though, right? We're looking for something we recognize, something we're familiar with.
Yeah, for sure. But all the research we've talked about today suggests that life could exist in environments that we would have thought were impossible. Like the extremophile paradox tells us that a planet or a moon doesn't have to always be hospitable to give rise to life.
So a planet that's had a lot of different environments in the past, even if it's pretty extreme now, could still have life. It's like habitability isn't a fixed thing, it changes over time.
Exactly. And looking at alternative biochemistry just expands our understanding of what's possible even more. We can't assume that water is absolutely necessary for life. We need to consider the possibility of life based on other solvents, different energy sources, even different genetic systems.
It's kind of humbling, isn't it? We're only just beginning to glimpse the true potential of life.
And that's what makes exobiology so exciting. It's constantly challenging what we think we know and reminding us that the universe is way more diverse and creative than we can even imagine.
This brings us back to how important it is to develop new ways to detect and study life beyond Earth. We need tools that can find a wider range of biosignatures, not just the ones we know from life on Earth.
Absolutely. And that's where things like nanopore sequencing are really important. Its ability to directly read genetic material, even those with those non-standard nucleotides, makes it a powerful tool for exploring all sorts of potential life forms.
It's like we're getting a brand new set of eyes to see the universe with.
And as we look through those eyes, we need to be ready for the unexpected. We might find microbes on Mars that are pretty similar to Earth life, or we might find organisms in the oceans under Europa's ice that completely change our understanding of what life is.
It's exciting, isn't it? The chance to find something truly alien, something that reshapes how we see life's role in the universe.
It reminds us that we're all part of this incredible cosmic experiment, a universe that's bursting with possibilities. And as we keep exploring, we need to approach it with humility, wonder, and a willingness to embrace what we don't know.
Well said. I think this deep dive has given us a lot to think about. We've explored the strange origins of extremophiles, the mind-blowing possibilities of alternative biochemistries, and how important it is to expand our idea of habitable. It's been a journey that's challenged our assumptions and sparked our imaginations.
It really has. And as we wrap up, I hope our listeners are leaving with a renewed sense of curiosity and a thirst for discovery. The search for life beyond Earth is just getting started, and the discoveries waiting for us have the power to completely change how we understand our place in the universe.
And who knows, maybe those discoveries will even inspire us to rethink how we view life here on Earth, to really appreciate its resilience, its diversity, and how interconnected it all is.
Definitely. In the end, searching for life beyond our planet is also about searching for ourselves. It's about understanding the nature of life itself and our place in the vastness of the cosmos.
This has been a really insightful deep dive. Thanks for sharing your expertise.
It's been my pleasure.
And to our listeners, keep exploring, keep asking questions, and keep looking up. The universe is full of wonders just waiting to be discovered.